Key points about lipid metabolism. All of your tissues need fatty acids for various purposes. This includes use as fuel, incorporation into membranes, production of steroids, or etc. But only your liver cells, so your like hepatocytes, and your fat cells or your adipocytes can make them. The liver and the adipocytes make fatty acids, store them as triacylglycerides or your tags, and ship them out to other tissues in need. These fatty acids have to be mobilized from triacylglycerols or your tag stores by lipases. And we'll talk more about, we'll review those in a second, but those include extracellular and intracellular lipases. Fatty acids are themselves made from acetyl-CoA, which are 2 carbons, and broken down to acetyl-CoA, as well as one propionyl-CoA, which is 3 carbons per odd chain fatty acid. The synthesis of fatty acids occurs mostly in the cytoplasm of liver and fat cells, and is a key user of NADPH, which can be made through the pentose phosphate pathway. I say it mostly happens in the cytoplasm of liver and fat cells. Some of it happens in the mitochondria, but mostly in the cytoplasm. Because acetyl-CoA can't get through the mitochondrial membranes, citrate and not acetyl-CoA is removed from the mitochondria to make fats. It is subsequently broken back down to acetyl-CoA by ATP-citrate lyase. So what we're looking at here, we take citrate out. We convert that to oxaloacetate using ATP-citrate lyase. Fatty acid breakdown occurs in most tissues and takes place mostly in the mitochondria in a process called beta oxidation. Very long fatty acids, so like greater than 20 carbons, are initially broken down via hydrogen peroxide mediated process in peroxisomes. But once they get to a shorter fatty acid, then those can be broken down in the mitochondria as well. The synthesis and the breakdown are reciprocally regulated. So key regulatory points are for the synthesis, acetyl-CoA carboxylase or ACC, which activates acetyl-CoA for incorporation. Remember, that was how we made our mobilized-CoA. And then that mobilized-CoA was going to inhibit the breakdown. Our key regulatory point for breakdown was carnitine acyltransferase 1, or CAT1, or CPT1, which allows our fatty acids into the mitochondria for breakdown. Remember, that was where we swapped that carnitine on in place for that CoA, and that was going to allow our fatty acids to enter the mitochondria, where they can be broken down through beta oxidation. Synthesis of fatty acids, however, that's happening in the cytoplasm. Remember, that's in our liver and our fat cells, and it uses NADPH, which we can make from the pentose phosphate pathway and from malic enzyme. This is mostly happening in your cytoplasm, a little bit in your mitochondria, but we're going to focus mainly on the cytoplasm. We mentioned how citrate, not acetyl-CoA, is removed from the citric acid cycle from the mitochondria in order to make fats, and broken back down to acetyl-CoA by ATP-citrate lyase. Now that you have acetyl-CoA in your cytoplasm, it's going to be pieced together by a multifunctional protein called your fatty acid synthase, or FAS. You're going to build 2 carbons at a time, but you're going to build them from a 3-carbon intermediate called mobilized-CoA. Mobilized-CoA was made through that ACC step through carboxylation. Mobilized-CoA is kind of the activated form because it has this beta-keto acid. Beta-keto acids are favorable to decarboxylate, and when they decarboxylate, the energy from that can be used to attack and add on to the growing fatty acid chain. This is a key site of regulation. It's going to be activated by citrate, an example of our feed-forward stimulation for an allosteric regulation, and inhibited by another type of allosteric regulation, this time feedback inhibition by palmitoyl-CoA. Mobilized-CoA itself is going to be an inhibitor of CAT1 or CPT1, which was involved in the fatty acid breakdown. Our ACC is also going to be regulated hormonally through phosphorylation and dephosphorylation, involving those hormones we see over and over when we're talking about metabolism, our insulin, that signal of kind of high energy, high glucose, that sort of thing, and its kind of counterpart, glucagon, our signal of low energy. And remember, glucagon is often working the same way as epinephrine. Epinephrine, that's kind of like our stress signal. So if we want to break down things for energy, we're going to have glucagon and epinephrine be acting. And if we want to store energy, we want insulin. Well, if we build fatty acids, we want to do that when we want to store energy, when we have a lot of energy. So insulin is going to promote the activation of ACC. And the way that it does it is by activating a phosphatase that dephosphorylates ACC. So what adds the phosphate group, the thing that you want when you want to inhibit the ACC. And so epinephrine and glucagon are able to act through pKa in order to phosphorylate the ACC and inactivate it so that you can use the acetyl-CoA for energy rather than building it up together. Other kinases also like AMPK, your signal of low energy storage status. In those cases, you're going to want to burn the acetyl-CoA for energy rather than build it up into fatty acids. So we have our covalent modification on top of the allosteric modification. This is going to allow for more like global regulation, sending signals throughout your body rather than just what's happening in that cell in that moment. For each cycle that we go through, we add 2 carbons and we use 1 ATP and 2 NADPH. The cycle kind of stops by default at 16 carbon saturated fatty acid called palmitate, which is cleaved off by FAS by the thioesterase subunit. Longer fatty acids and unsaturated fatty acids can be made via elongation and desaturation on the ER. One note, though, is that although we can add double bonds, we can't do so past the 9-10 position in our delta nomenclature. So when we're thinking about counting, starting from the carboxylic acid side, we can't add a double bond past the position between carbons 9 and 10. What this means is that, practically what this means is that we can't introduce an omega-3 or omega-6 double bond. So omega notation that counts from the other end. So from kind of just the methyl, the alkane end. What that's going to mean is that if you want an omega-3 or an omega-6 fatty acid, you're going to have to get that through your diet. You can then modify it to get more complicated and longer and stuff, omega-3 and omega-6. But because you're adding, when you add to the delta end, not the omega end. The omega position is not going to change when you elongate a fatty acid. And so if you can't introduce that bond at that omega-3 or omega-6 position, you can't transform another fatty acid that doesn't have one of those into one of those. So look at the other video if you want to know more about that. But those kind of the elongation and the desaturation, that's going to happen in like the membrane of your ER. Okay, fatty acid catabolism. This is going to occur in your mitochondria via beta oxidation. We talked about how CAT1 or CPT1 was the key regulator of that. We're going to be breaking down fatty acids 2 carbons at a time. For each cycle, we're going to be cutting off an acetyl-CoA, producing an NADH and an FADH2 that can be used to make ATP. If we have odd-chain fatty acids, that's going to leave us also with the 3-carbon propionyl-CoA, which gets converted to succinyl-CoA, which can be used in the TCA. That's going to be an example of an anaplerotic reaction. It can replenish intermediates if we take intermediates out to do things like, say, make glucose. Therefore, we say that odd-chain fatty acids can be glucogenic, so we can make glucose from them, but not the even-chain fatty acids. That doesn't mean that you can't find, say, carbons from your fats inside of your glucose. It just means that if all you had were fatty acids, they couldn't be used to sustainably make glucose because they can't enter the TCA unless oxaloacetate is already present, and oxaloacetate wouldn't be present on net if you were taking out a malate for every like acetyl-CoA you brought in. If you took out a malate to make glucose, that wouldn't work. Before we can actually do beta oxidation, you have to invest some energy and sneak those fatty acids into the mitochondria. The activation happens through fatty acyl-CoA synthetase or ligase, is ACS. What it does is it's going to spend ATP, the equivalent to spending 2 ATP, in order to attach a CoA to your fatty acid. That leaves you with the fatty acyl-CoA. That fatty acyl-CoA is what is a starting product for, or starting material for beta oxidation, but that beta oxidation happens in the mitochondria and we're still in the cytoplasm. So we're going to have to go through some trickery in order to bring it in. The first thing we got to do is we're basically going to use this carnitine shuttle, where the fatty acid still in the cytoplasm is handed from CoA to carnitine by carnitine acyltransferase 1, or CAT1, that key regulatory step. The fatty acid now attached to carnitine is then transported into the mitochondrial matrix by carnitine acyl-carnitine translocases, which also brings the carnitine back to the cytoplasm. Once you are in your mitochondria, now we can reverse the situation using Cat2, and the fatty acyl-CoA can now be broken down by beta oxidation. So we have this swap off from we have this activation, attach a CoA, swap that CoA for a carnitine, bring that in, bring that through, convert it back to our fatty acyl-CoA, and now we're ready for beta oxidation. That beta oxidation would generate a lot of acetyl-CoA. If that acetyl-CoA can enter the TCA, all is great. If it can't, however, it builds up. And that last step of beta oxidation, that thiolase step, reverses itself, followed by a couple of other enzymatic steps that results in the formation of ketone bodies. This can happen in the case of diabetes if there's not enough oxaloacetate to keep the TCA running. It can happen in the case of ethanol intoxication because the NADPH builds up from ethanol oxidation and inhibits the TCA. Or it can happen in starvation because you don't have enough oxaloacetate and you have excessive breakdown of ketogenic amino acids. What can happen if you have a lot of ketone body formation is that it can acidify the bloodstream, causing a condition called ketoacidosis. But before you get away thinking that all ketone bodies are bad, they're really important for your brain because since they're soluble, they can go through like the blood-brain barrier, whereas your lipids, your fatty acids that are traveling through your bloodstream on like serum proteins, those can't actually get to your brain readily. So ketone bodies are able to offer a quicker source of fuel for your brain, allowing it to have energy to keep going under conditions where glucose is low. Your brain can't like store glycogen and stuff, so it really relies on those ketone bodies if it needs energy. And those ketone bodies can also be formed, say, from some amino acids. In speaking of fat mobilization, cells take up fatty acids and not triacylglycerols for use as fuel, but fats as energy are stored as these triacylglycerols. In order to use the fatty acids, break down the fatty acids, they need to be cleaved off of the glycerol backbone by enzymes called lipases for uptake and subsequent use. Fatty acids are delivered from liver cells to tissues packaged as tags, bundled up with phospholipids, cholesterol, and other hydrophobic stuff in the interior of a lipid-coated bubbles called lipoproteins. The fatty acids, before they're taken into the cells, they're broken off of the glycerol backbone by lipoprotein lipase on the surface of blood vessels. That was happening when your liver was sending out fatty acids. So your liver was sending them as tags in these lipoproteins. The fatty acids that are delivered from your adipocytes, so your fat cells, are actually delivered as fatty acids, not on lipoproteins. But because fatty acids aren't water soluble, unlike those ketone bodies, they're going to have to travel kind of piggybacking on proteins, typically your serum albumin protein, which kind of just has these globby hydrophobic surfaces that can glob onto fatty acids, as well as bind non-specifically to various drugs and stuff, which can either sometimes like slow the uptake of various drugs, can sometimes be good, can sometimes be bad. But these fatty acids then can travel through your bloodstream and reach tissues. They don't need to be cleaved off because they were already cleaved off inside of your adipocytes. So how are your adipocytes going to cleave them off? That's going to happen intracellularly by hormone-sensitive lipase. The hormone-sensitive lipase is sensitive to the hormones adrenaline and glucagon. So if you have low blood sugar, if you have low energy, your fat cells are that's going to activate the hormone-sensitive lipase, break down the fats, send those fatty acids out into the bloodstream where they can be bind to the serum albumin and be taken to cells in need. This helps us mobilize fuel stores for breakdown for energy inside of the shell or the shipping out into other cells. So there you have it, a whirlwind tour of our lipid metabolism.